Welcome to BioTales, where we get into the stories behind the science shaping biotech. Today, we're heading over to Oxford in the UK to look at a company called Sitryx Therapeutics.
Sitryx. They have a really ambitious goal, I think it's fair to say.
Definitely. They're aiming to, rebalance the immune system, and they're doing it by rewiring its metabolism, essentially.
It's a fascinating approach. We'll be exploring their work in this field, immunometabolism. It's, generating quite a bit of excitement.
Absolutely. So let's set the scene. Where does the Sitryx story really kick off?
It properly gets going around the summer of 2018. And this wasn't your typical startup. It was more like a convergence, really. 6 very well-known scientists, all leaders in their fields, came together.
Okay, 6 heavy hitters. Who are we talking about?
Well, you had Homan Ashrafian. He's a venture capitalist, also a cardiologist. Yeah. Played a key role getting the seed funding. Then, on the academic side, people like Luke O'Neill, the Irish immunologist, known for his work on inflammasomes. Really big name. And Jonathan Powell from Johns Hopkins, an expert in T-cell metabolism. Jeff Rathmel at Vanderbilt, he's often called one of the fathers of immunometabolism.
Wow, okay.
Michael Rosenblum from UCSF, focusing on regulatory T-cells in the skin. And Paul Petertok, a rheumatologist who previously headed up immunoinflammation R&D at GSK.
So a real powerhouse team. What was the big vision that brought them all together?
They shared this pretty audacious vision, creating a completely new class of drugs.
Not just another anti-inflammatory.
Not at all.
No.
The idea wasn't just suppressing inflammation in the, let's say, conventional way. It was about subtly kind of steering the metabolic engines inside immune cells.
Steering them.
To correct the underlying immune imbalances, a deeper level of control.
That's fascinating. And you mentioned Paul Petertok coming from GSK. Was there a connection there? How did this collaboration actually happen?
Yeah, that's a really interesting part of the story. It's not just about the science, but the human element, the collaboration.
Right. And GSK played a role in that.
A key role, actually, through their internal immunology network program. It was, frankly, a brilliant strategic move by GSK.
How so?
Well, it was designed specifically to de-risk that very early stage innovation, to fast-track academic breakthroughs into potential commercial ventures. It really changed the dynamic between big pharma and academia.
Okay, so what did this program actually do?
It funded sabbaticals for leading academic immunologists, embedded them right inside GSK's R&D hub in Stevenage for up to 3 years.
So they're inside the corporate R&D environment.
Exactly. But they could continue their own independent research. The key was they were working day-to-day alongside GSK scientists, using GSK platforms, getting access to their tech, their IP. It was all about sparking new ideas, new translational projects. Open innovation, really.
Okay, so that's the fertile ground. But how did Sitryx, the company itself, emerge from that? Was there a specific aha moment?
There kind of was, yeah. It's almost a classic biotech origin story moment. The actual concept for Sitryx started taking shape during these, informal meetings and chats at GSK.
Yeah.
Luke O'Neill was there on his sabbatical.
Right.
And he's just talking with Paul Petertok, who was running the whole immunology network program. And they start kicking around this idea. Could we actually reprogram immune cells by tweaking their metabolism?
Just like that.
Well, the specific spark, the idea that really ignited things, was when they discussed how certain metabolites, succinate and aconate, could potentially reprogram immune cells. That was the moment. O'Neill even joked later that his sabbatical turned out to be way more productive than he'd planned.
I bet.
That realization pushed them to gather the other key metabolism researchers, the ones we mentioned, and form a company focused purely on translating these insights into real drugs.
That really does sound like an organic way for a company to start. And the funding story seems to back that up. Started with seed capital?
Yep, seed capital from SV Health investors right there in Oxford. Then, within about 2 years, they'd brought in partners like Sofinova. GSK itself actually invested, Oxford Science Enterprises too.
Okay, building momentum.
But the really big one early on was striking that exclusive collaboration with Eli Lilly.
Right, potentially worth up to $570 million.
Exactly. For a company that new, getting that level of commitment from a major player like Lilly, it tells you a lot about the perceived scientific promise, doesn't it?
It really does. It's a massive signal. So what was attracting these investors so strongly?
I think it boiled down to 2 main things. First, just the sheer collective track record of the founders. I mean, between them, they'd published over 1,000 papers on immunometabolism.
1,000? That's incredible.
And second, that early access they had to GSK's chemistry capabilities and intellectual property, thanks to Tox Immunology Network. That significantly lowered the usual venture risk.
Right. It de-risked it early.
This unique setup, top academics, a focused VC-backed startup, and a big pharma partner, all aligned. Let them raise more funding relatively quickly. By 2023, they were over $75 million raised, plus that Lilly deal solidified.
There must have been serious excitement.
Oh, definitely.
So what was the absolute core belief driving these founders? What was the fundamental idea?
Their core conviction was, well, pretty revolutionary at the time. They argued that chronic autoimmune diseases aren't simply overactive immunity.
Okay.
Instead, they believe these diseases happen because immune cells get fundamentally stuck in the wrong metabolic gear, like an engine running inefficiently or just plain wrong for the conditions.
Stuck in the wrong gear.
Yeah. And so the thinking was, if you could find a way to precisely flip those metabolic switches back,
you could potentially fix the problem at a deeper level.
Exactly. Not just dampen the symptoms, but actually restore immune tolerance, get the system back into balance, and maybe achieve actual disease remission. It's a paradigm shift from just managing inflammation.
Okay, that's a really compelling vision. Before we dive into how Sitryx plans to flip those switches, let's maybe take a step back. We've used the term immunometabolism a few times. Can we unpack that a bit? What exactly is it?
Sure. Yeah, it's a field that's really taken off. At its core, immunometabolism is the study of how metabolic pathways, the processes cells use to get energy and building blocks.
Like glycolysis, the Krebs cycle.
Precisely those. Glycolysis, Krebs cycle, fatty acid oxidation, the pentose phosphate pathway. How these processes inside immune cells directly control or dictate what those cells become, how they behave, and what their ultimate function is in an immune response.
So the cell's metabolism determines its job, essentially.
In many ways, yes. Think of it like the fuel choice and the engine tuning for different types of immune cells. For instance, those pro-inflammatory effector T cells, the ones that mount an aggressive attack.
Right, the frontline fighters.
They rapidly switch to using anaerobic glycolysis. It lets them generate energy, ATP, very quickly, even if it's not the most efficient long term. They're like race cars, burning fuel fast for immediate power.
High octane, quick burn. Got it. How does that compare to other immune cells, then? The ones involved in calming things down or remembering?
It's quite different. Take regulatory T cells, or memory T cells, the ones crucial for maintaining tolerance, preventing autoimmunity, and remembering past threats. They tend to rely much more on oxidative phosphorylation and fatty acid oxidation.
So more like fuel-efficient engines.
Yeah.
Filled for the long haul.
That's a great analogy, exactly. They're geared for endurance and controlled responses. The really key point here is that these different metabolic programs aren't just byproducts. They actively determine whether the overall immune response is going to be aggressive and inflammatory or tolerant and regulatory. It's a fundamental control layer.
Okay, so that's how it should work. A balance of different metabolic states for different jobs. What goes wrong in autoimmune diseases? How is that balance disrupted?
That's where the problem lies. In autoimmune conditions, this metabolic balance gets seriously skewed. Immune cells, particularly effector cells, get stuck in that pro-inflammatory, high glycolysis metabolic state.
The race car mode gets stuck on.
Kind of, yeah.
Mm-hmm.
And that drives chronic inflammation and leads to the tissue damage we see in these diseases. There's some specific examples from the founders' work that really underpin this, like Luke O'Neill's group finding that in activated macrophages, a metabolite called succinate builds up, and this actually enhances inflammation. But then, another metabolite they studied, itaconate, does the opposite. It actively counters the inflammation.
Interesting, like a metabolic push and pull.
Exactly. And then Jeff Rathmel showed just how essential glycolysis is for T cells to proliferate, to multiply rapidly. And crucially, he showed that tweaking a specific enzyme involved, called PKM2, could actually suppress autoimmunity in models.
PKM2, we'll come back to that one.
We will. And Jonathan Powell's lab identified another enzyme, MTHFD2, which is in the mitochondria. They found it acts as a critical checkpoint controlling the proliferation of those effector T cells. And inhibiting MTHFD2 could actually encourage the development of regulatory T cells instead.
So these specific discoveries about succinate, itaconate, PKM2, MTHFD2, these are the actual scientific foundations for Sitryx's approach.
Absolutely. They form the bedrock. They provide the specific molecular targets.
Okay, so taking all that background, what's the really big why it matters here? Why is targeting these metabolic enzymes potentially such a game changer compared to, say, traditional immunosuppressants like steroids or even some biologics?
I think the core insight, the real elegance of the approach, is about precision and resetting the balance, instead of just hitting the immune system with a broad hammer.
which can have side effects like increased infection risk.
Exactly. By selectively targeting these metabolic enzymes that are particularly active or upregulated specifically in the pro-inflammatory effector cells, the theory is you can gently nudge the whole system back towards regulation, back towards tolerance.
A nudge, not a hammer.
Precisely. It offers the potential for a much more targeted intervention, aiming to restore the natural balance without causing that widespread immunosuppression that makes patients vulnerable. It's about retuning, not just silencing.
Okay, that makes a lot of sense. Let's talk about how Sitryx is actually trying to build this tuning kit, their product engine. How do they go about finding and hitting these targets?
It starts with really meticulous target discovery. Their teams systematically map out and scan the metabolic pathways inside activated B and T cells.
What are they looking for specifically?
They're hunting for enzymes that meet two criteria. One, they have to be absolutely critical for that pro-inflammatory function for driving the disease. Two, and this is just as important, they should ideally be dispensable or less important for the normal function or resting immune cells or those beneficial regulatory cells.
Finding that specific vulnerability.
Exactly. Find that key node, that metabolic knob you can turn. Once they identify a promising enzyme target, then the chemists step in. Their job is to design small molecules, drugs you can likely take as a pill, that can precisely modulate that enzyme's activity.
Modulate meaning activate or inhibit?
Either. It depends on the target. Sometimes you want to boost an enzyme's activity, sometimes you want to block it. It's about designing a molecule that fits perfectly and turns that specific metabolic switch up or down as needed.
Right, the magic of drug design. So you mentioned PKM2 earlier, based on Jeff Rathmel's work. That became one of their key targets, didn't it? Tell us more about that one.
Yes, PKM2 is a really fascinating target. It's more than just a metabolic enzyme. It really acts like a master switch inside immune cells, particularly T cells.
A master switch? How?
Well, normally PKM2 helps process glucose, but when immune cells get activated in an inflammatory context, PKM2 can change its form. Instead of just staying in the cytoplasm doing this energy job, a less active form actually moves into the cell nucleus.
Into the command center.
Right, into the command center. Yeah. And once it's there, it starts influencing gene transcription, basically. Fleming genetic switches that drive inflammation, promote cytokine production, and push T cells towards those pathogenic Th1 and Th17 types that cause so much trouble in autoimmunity.
So it links metabolism directly to inflammatory gene expression.
Precisely. This PKM2-driven metabolic reprogramming is a key step in differentiating those harmful T-cell subsets.
Okay, so if PKM2 is doing that, how does Sitryx target it? What's the strategy? Is it specific enough?
That's the crucial part. The strategy isn't to just block PKM2 entirely. Instead, they're developing pharmacological activators.
Activators. That sounds counterintuitive if it promotes inflammation.
It does at first glance. But the idea is to develop molecules that lock PKM2 into its fully active tetrameric state, its normal energy processing form in the cytoplasm.
Ah, so you keep it busy doing its day job so it can't moonlight in the nucleus, causing trouble.
That's a great way to put it. By keeping it locked in that active state, you prevent that shift. You prevent it from going into the nucleus and driving those inflammatory genes. You keep it focused on efficient energy metabolism.
Okay, that makes sense. And does it work?
Well, in preclinical models of autoimmune diseases, this strategy activating PKM2 to keep it cytoplasmic has shown promising results. It reduced T-cell proliferation, lowered those key inflammatory cytokines like interferon gamma and IL-17 from Th1 and Th17 cells, and ultimately suppressed disease severity in those models.
And Sitryx has a drug based on this?
Yes, their lead candidate here is SYX5219. It's an oral PKM2 activator designed specifically to do this. Rebalance B and T cell metabolism, dampen those inflammatory responses. They're initially looking at it for conditions like atopic dermatitis. And this was a huge step for them. In April 2024, they officially nominated SYX5219 as their very first clinical candidate.
That's a major milestone.
Huge. And things moved quickly after that. Just this past March 2025, they announced they started the first in-human phase 1A trial in healthy volunteers. And the plan is to move into a phase 1B study in patients with moderate to severe atopic dermatitis later in 2025.
So we should start getting clinical data relatively soon. It's really rapid progress from nomination to clinic.
That's definitely one to watch. What about other targets? You mentioned MTHFD2 earlier from Jonathan Powell's work.
Yes, MTHFD2 is their next major target moving forward. It's another enzyme, but this one is located in the mitochondria, the cell's powerhouses. It's involved in something called 1-carbon metabolism. Okay, what does that pathway do?
It provides essential building blocks, particularly purines and thymidylate, which cells need to make DNA and RNA, especially when they're dividing rapidly.
Like activated immune cells.
Exactly. And what Powell's lab and others found is that MTHFD2 is significantly upregulated, meaning its levels increase a lot, specifically in activated Th1 and Th17 cells, those pro-inflammatory ones again.
So it's fueling their rapid expansion.
Right. And the really key finding one Sitryx is leveraging came from Powell's pivotal study. They showed that if you either genetically remove MTHFD2 or inhibit it with a drug,
you starve those pro-inflammatory cells.
Essentially, yes. You deplete the building blocks they desperately need to proliferate. But there was a fascinating, almost unexpected consequence. This metabolic stress didn't just stop the bad cells. It actually seemed to promote the differentiation of regulatory T cells, the good guys that help maintain tolerance. It shifted the balance by promoting FOXP3 expression, which is the key marker for Tregs.
So inhibiting MTHFD2 could be a double whammy, suppress the inflammatory cells, and boost the regulatory ones.
That's the therapeutic hypothesis, exactly. And it's supported by observations that MTHFD2 levels are elevated in tissues from patients with various autoimmune diseases, ulcerative colitis, rheumatoid arthritis, psoriasis.
Makes sense. So Sitryx has an inhibitor for this.
They do. Their drug candidate is called SIT-047. It's designed as a first-in-class selective MTHFD2 inhibitor. The goal is to selectively hit those Th1, Th17 responses while hopefully sparing or even boosting the regulatory T cells. In May 2025, just a couple of months ago, Sitryx announced SIT-047 and their initial target indication, psoriatic arthritis.
Psoriatic arthritis affecting skin and joints. Nasty condition.
Very much so. They've reported potent efficacy in preclinical models, and the plan is to submit for regulatory approval to start clinical trials in the first half of 2026.
So another one moving towards the clinic fairly soon. It really shows the breadth they're building.
It really does. Having two distinct novel mechanism candidates like SYX5219 and SIT-047 moving forward underscores their ability to translate that deep immunometabolism science into potential drugs.
Beyond these two internally developed programs, there's also that big Eli Lilly partnership you mentioned earlier, focusing on an itaconate mimetic, SYX1042. That came from Luke O'Neill's work, right?
That's right. The itaconate story is another fascinating thread. The licensing deal itself, back in 2020, was obviously a massive milestone and validation for Sitryx. Lilly saw the potential in immunometabolism and committed significantly up to $570 million in potential milestones.
And Lilly took on the development.
Yes. Under that deal, Lilly took the reins for developing SYX1042. To understand what it does, you need to understand itaconate.
Okay, so what's special about itaconate?
Itaconate is a metabolite that our own immune cells, specifically macrophages, produce naturally when they get activated. Luke O'Neill's lab did seminal work figuring out what it does. It acts as a natural brake on inflammation. One key way it works is by modifying a protein called KEAP1. When KEAP1 is modified by itaconate, it releases another protein it normally holds onto, called NRF2. NRF2 is a master regulator, a transcription factor. When it's released, it travels to the nucleus and switches on a whole suite of powerful antioxidant and anti-inflammatory genes.
So itaconate triggers the cell's own defense system.
Exactly. Plus, itaconate also directly inhibits other inflammatory pathways like succinate dehydrogenase and the NLRP3 inflammasome, further reducing the production of inflammatory cytokines like IL-1 beta.
So SYX1042 is designed to mimic that action.
Precisely. It's an itaconate mimetic designed to activate that NRF2 pathway, switch macrophages towards an anti-inflammatory state, and provide broad antioxidant effects, a multi-pronged anti-inflammatory approach based on a natural mechanism.
Clever. So what happened with the Lilly partnership in this drug?
Well, Lilly moved it forward. They started a phase 1 trial of SYX1042 in January 2024. According to Sitryx, that first in-human phase 1 study in healthy volunteers was completed successfully. They reported positive first-in-human data.
Sounds good. So moving to phase 2 with Lilly.
Actually, no. This is where it gets interesting. The program has since returned to Sitryx.
Returned? Why?
Apparently, Lilly decided to reprioritize its internal pipeline, specifically within pain and inflammation R&D. So they decided to return the rights to Sitryx after completing that phase 1 study.
Was there a problem with the safety?
Sitryx has been clear that wasn't the reason. They state it wasn't due to any safety signals or pharmacokinetic issues. It seems to have been purely a strategic portfolio decision by Lilly.
So Sitryx gets it back. What state is it in?
They get back a phase 2 ready asset. It comes with that positive phase 1 data package already validated in humans. So while the partnership ending might seem like a setback, Sitryx now has regained full rights to a clinically de-risked, promising compound.
That's actually a pretty significant outcome for them. Gives them another later stage option.
A very significant return, yes.
And it's not just these 3 assets, is it? They have others in earlier stages, too.
That's right. Their pipeline is broader. They also mentioned a glutamate 1 inhibitor they're exploring for allergic asthma.
Targeting glutamine metabolism.
Yes. And another one, which is a salt-inducible kinase 2, or SIK2 inhibitor. They're looking at that one for ulcerative colitis.
So multiple targets, multiple pathways, multiple potential indications.
Exactly. It really illustrates their overall strategy. Trying to build, as they put it, a whole menu of metabolic knobs. Different ways to fine-tune the immune system by targeting its metabolic wiring.
That menu analogy makes sense. Which brings us back to their journey, the milestones and partnerships that got them here. You mentioned the leadership evolved, too.
Yes, leadership evolution often reflects a company's stage. Neil Weir, who had a strong discovery background from UCB, was the founding CEO and did a great job getting things established. He's now president.
And the new CEO.
In July 2024, Ian Kilty took over as CEO. He brings extensive clinical development and commercial experience. That shift really signals the company's increasing focus on clinical execution, getting these drugs through trials.
Makes sense as the pipeline matures.
Definitely. And they also brought in Ravi Rao as Chief Medical Officer. He seems very focused on keeping the patient perspective central, really emphasizing the high unmet need in diseases like atopic dermatitis and psoriatic arthritis, which are their first clinical targets.
So putting it all together, Sitryx seems to be at a really interesting point right now, an inflection point maybe.
I think that's exactly the right term. On the one hand, you have this incredibly strong scientific rationale. We've talked about it. Metabolic pathways are fundamental. They're conserved. They directly control immune cell fate. It's built on solid biology.
There's always a but in biotech.
There usually is. The but here, the major challenge defining this inflection point, is that no immunometabolism drug has actually made it all the way to approval yet. Not for autoimmune disease.
So the ultimate clinical validation is still missing for the whole field.
Precisely. Which means the upcoming clinical data for Sitryx is absolutely crucial, not just for them, but arguably for the field. Those phase 1 readouts and especially the subsequent phase 2 results for SYX5219 and SIT047, they will be make or break.
They have to show it works in people.
They have to show that modulating PKM2 or MTHFR2 actually provides a tangible clinical benefit to patients safely. This is the moment the elegant bench science has to translate to the bedside. Everything hinges on that clinical proof of concept.
Given that high-stakes challenge, what really sets Sitryx apart? What are their key differentiators in this emerging, still unproven space?
They do have several advantages, I think. First, as we've discussed, is the breadth of their pipeline. They're not a one-trick pony. They have multiple shots on goal targeting different metabolic nodes.
Diversification.
Right. Second, and this is potentially very important, is the specificity of their targets. The enzymes they're going after, like PKM2 and MTHFR2, seem to be selectively upregulated primarily in the activated disease-driving immune cells.
Why is that so important?
Because that specificity holds the promise of limiting off-target effects, potentially leading to better safety and tolerability compared to broader immunosuppressants. That's a major potential advantage.
And the team.
And finally, yes, the team. You can't discount the foundation laid by those world-class scientific founders, true pioneers in immunometabolism. Combined now with an experienced, industry-savvy management team focused on clinical translation and development, that's a powerful combination.
But even with those strengths, they're not alone in this space anymore, are they? The competition must be heating up.
Oh, absolutely. The whole field of immune modulation via metabolism is attracting a lot more attention now. You have other emerging biotechs, and you can bet the big pharma companies are exploring this area too, either internally or through partnerships.
So speed matters.
Speed and execution, definitely. Sitryx needs to move efficiently, generate convincing safety and efficacy data quickly to really solidify its position and maintain that early lead. It's definitely becoming a more competitive landscape.
Looking further ahead then, let's assume they are successful. If these programs make it through, how could that change the future of treating autoimmune disease?
If they succeed, it could be truly transformative. We could see an era where metabolic modulators become a standard part of the treatment toolkit for autoimmune diseases, right alongside biologics and other therapies.
A whole new class of drug.
A whole new therapeutic modality, yes. It would represent a major paradigm shift. It's interesting to draw a parallel with oncology.
How so?
Oncologists are already using metabolic inhibitors as part of cancer treatment. They exploit the altered metabolism of cancer cells. For instance, there are drugs targeting lactate transport or mitochondrial metabolism or even arginine metabolism in the tumor microenvironment.
Concepts influenced by immunometabolism research itself.
Very much so. There's a lot of crosstalk between cancer metabolism and immunometabolism. So if Sitryx can prove this works for autoimmunity, it would bring that same kind of targeted metabolic intervention into rheumatology, dermatology, gastroenterology.
Expanding the options significantly.
Hugely expanding the options. And crucially, it offers the hope of moving away, at least partially, from relying so heavily on broad immunosuppression, giving patients therapies that are potentially more precise, better tolerated, and maybe even capable of restoring that underlying immune balance we talked about.
It's a really compelling future vision. As we start to wrap up our exploration of Sitryx today, what does their story tell us about biotech innovation more broadly?
I think Sitryx is a fantastic case study, really. It exemplifies how deep, fundamental, basic science discoveries, understanding the roles of molecules like succinate, atacanate, PKM2, can genuinely inspire entrepreneurs to think completely differently about treating disease, to create entirely new therapeutic approaches from scratch.
And it highlights the power of collaboration, too.
Absolutely. It's a perfect example of that synergy you need. World-class academic science, savvy venture capital backing, and strategic input and resources from big pharma like the early GSK connection and the Lilly deal. Getting all those pieces working together is often key to success.
It really underscores the core promise of immunometabolism itself, doesn't it?
It does. That central idea that by meticulously rewiring the energy pathways of immune cells, we might be able to do more than just manage symptoms. We might actually get closer to restoring true, durable immune balance. It's an incredibly exciting prospect.
So for everyone listening today, particularly researchers and investors active in this space, the message seems clear.
Keep a very close watch on Sitryx's clinical data over the next year or two. Those results, particularly from the Phase 1b and subsequent Phase 2 trials for SYX5219 and SIT047, will be pivotal. They'll be the real test of whether immunometabolism is ready to make the leap from compelling scientific concept to validated clinical reality for autoimmune disease.
The potential is huge, but the proof is still needed. For now, though, the Sitryx story is a powerful reminder. It shows that really bold ideas, when they're backed by rigorous science and guided by experienced leadership, have the genuine potential to reshape therapeutic landscapes.
Well said. It's a journey worth following.
Indeed. Thanks for joining us on BioTales today. If you found this look into Sitryx and immunometabolism valuable, please do subscribe and share the show with your colleagues.
And we'll be back next time, exploring another company, pushing the boundaries of biotech innovation.
See you then.